Welcome back to the deep dive. Today, we are right on the edge of something. Well, something truly transformative in drug discovery.
We really are.
We're looking at this incredible convergence of deep learning, atomic-level biology, and, the future of medicine, all happening at once.
It's moving so fast.
And it makes you wonder. The old ways of finding drugs, these slow, generalized screening methods, they feel like they're on notice. So the big question we're really digging into is, how close are we really to a world where AI doesn't just help design drugs, but actually creates these complex, bespoke molecules from scratch with totally predictable results?
And that's exactly the mission for today. To get at that answer, we've pulled insights from two really critical sources of these breakthroughs.
Okay, so what are we looking at?
On the one hand, the technology itself. We're going deep on a new DL pipeline called RFpeptides. It's built to tackle one of the toughest challenges out there, designing therapeutic macrocycles.
Right, these closed-loop molecules.
Yeah.
And then on the other hand, the application.
Exactly. We'll look at the clinical frontier, focusing on these next-gen GLP-1 co-agonists. The drugs making huge waves in treating metabolic diseases like type 2 diabetes and obesity.
So the goal is to connect the tech to the therapy.
That's it. We want to show how these computational leaps are taking us from just slow, random searching to incredibly fast, precise, custom-designed therapies. It's about shortening that path from an idea to a potential drug.
A shift from stochastic discovery to deterministic design. I like that.
That's the phrase.
Okay, let's unpack that. In our first segment, let's focus just on the tech. RFpeptides. The traditional challenge with macrocycles, these special peptides, was that you had to just screen enormous libraries, right?
Enormous is an understatement. Macrocycles are fantastic drug candidates. They're stable. They can get into cells better than linear peptides. But making them, it's been brutally difficult.
Why so hard?
Well, the traditional methods, high-throughput screening, building these huge chemical libraries, it's incredibly resource-intensive. And even when you get a so-called hit, you have almost no control over how it actually binds to the protein you're targeting.
So you find the needle in the haystack, but you have no idea which end is sharp.
Precisely. You might have to screen, and this is not an exaggeration, trillions of unique peptides just to get a few starting points. The whole process was limited by the sheer size of chemical space.
And this is where generative AI comes in. RFpeptides is described as a denoising diffusion-based pipeline. For anyone following this space, that sounds a lot like the tech behind tools like RosaFold or RFdiffusion.
It's the same family of technology, absolutely. They've adapted it. The basic idea with diffusion models is you start with random noise, total chaos, and the model learns to denoise it into a coherent structure.
But doing that for a closed-loop peptide, a macrocycle, that must have been a unique challenge.
A huge challenge. For one, there isn't a ton of training data. And second, how do you mathematically force the AI to make a closed loop every single time?
Right. How do you tell it, hey, the beginning has to connect perfectly with the end?
That's the secret sauce, the big innovation. The RFpeptides team extended the RosettaFold 2 network with a novel method they call cyclically symmetric relative position encoding.
Okay, that's a mouthful. What does that actually mean in practice? What's the trick?
Think of it like giving the AI a non-negotiable architectural rule before it even starts.
Yeah.
Normal models just predict where atoms are relative to each other. This encoding forces the model to treat the start and the end of the peptide chain as the exact same point.
Ah, so it's a constraint.
A built-in constraint. It's telling an AI, whatever you design, the first atom and the last atom must be in a position to form a bond. It imposes the macrocycle shape from the get-go.
So instead of making a string and hoping it can form a ring,
It generates a ring by design. It's deterministic engineering, not just a search. And the efficiency numbers, because of this, they're just astonishing.
how astonishing.
Instead of screening trillions of molecules, they got medium to high affinity binders for 4 different protein targets, including big cancer targets like MCL1 and MDM2 by testing. Get this, 20 or fewer designed macrocycles per target.
20, down from trillions. That's a different reality for drug discovery. Lead generation can now happen at the speed of a computer, not a giant lab.
It completely flips the economics. You spend your resources designing a small, high-confidence set, not blindly searching a vast one.
That predictability is the perfect lead into our next segment, structural fidelity. Because designing a molecule is one thing, but if the computer model doesn't match the real thing, You're back to square one.
Yes, and this is where it gets really powerful. The reason atomic accuracy by design is so revolutionary is that it completely obliterates the structure determination bottleneck.
Explain that bottleneck.
In the old workflow, you find a hit. Then you spend months, maybe years, trying to use X-ray crystallography or cryo-EM to figure out exactly how it binds. Only then can you start trying to make it better.
But with RFpeptides,
You get that binding information up front. The design is the binding mode, so researchers can jump straight to optimizing the drug candidate.
and they proved this experimentally. The X-ray structures confirmed the designs. Let's talk numbers. How strong was the binding and how close was the match?
The binding was incredibly strong. For two of the targets, GABARAP and RBTA, they got binders with dissociation constants. That's K sub D of less than 10 nanomolar.
For our listeners, a low KD is good.
Very good. It's a measure of how sticky the molecule is. A KD of 6 nanomolar, which they got, is like molecular superglue. It binds to the target and it does not let go. That's a sign of a very potent drug.
Okay, that's the potency. What about the accuracy? The structural match?
This is where the engineering is just beautiful. The X-ray data show that the computational models matched the real-world structures with an RMSD that's root mean square deviation between 0.7 and 1.4 angstroms.
So an angstrom is, what, 1/10 billionth of a meter?
Yeah.
Put 1.0 angstrom of error into context for us.
A typical carbon-carbon bond is about 1.5 angstroms long. So an RMSD of 1.0 means the average difference between the predicted atom positions and the real ones is less than the length of a single chemical bond.
That is truly atomic precision. The AI didn't just find a key that fits the lock. It designed the key's exact shape down to the individual groove.
That's it. And then there's the RBTA case study, which might be the most important part of this whole story.
Why is that?
Because for that target, RBTA, they didn't even start with an experimentally solved structure. They started with a structure that was predicted by another AI, like AlphaFold 2.
Wait, so the AI predicted the lock and then another AI designed the key for that predicted lock.
Exactly, and it worked. This demonstrates a true end-to-end AI-driven drug discovery pipeline. Historically, if you didn't have a high-resolution structure of your protein target, it was basically considered undruggable.
But this means we can now start designing drugs for potentially thousands of proteins that we only know about from their genetic sequence. It just blew the doors off the druggable universe.
It expanded the map overnight.
Okay, that is incredible. And it all relies on being able to validate these structures. So let's zoom out a bit in segment 3 and talk about that toolkit. How do X-ray crystallography and cryo-EM fit into this new world?
Well, X-ray has been the gold standard for decades, and it's what they use to validate these RFpeptides. It's perfect for getting super high-resolution pictures of rigid things, but it requires you to grow a perfect crystal of your protein, which can be a huge roadblock.
The biology bottleneck.
Yeah.
And that's where cryo-electron microscopy, cryo-EM, really had its moment, especially since around 2012.
A total revolution. It was driven by new hardware, these direct electron detectors, and incredibly sophisticated software like Reliance. Suddenly, you could get near-atomic pictures of molecules just by flash freezing them in a thin layer of ice.
Oh, crystals needed?
No crystals. And that's why it's so essential for the kind of drug targets we're going to talk about next. Its real strength is solving structures of huge, complicated molecular machines.
Things that don't crystallize well.
Or things that are really flexible and dynamic. X-ray gives you a static snapshot. Cryo-EM can capture a molecule in different functional states. It can see it move.
You mentioned the ryanodine receptor. RYR1 is a classic example of this.
Yes, RYR1 is this massive calcium channel, just enormous. People tried for years to crystallize it with no luck. Cryo-EM solved its structure, no problem. And for drug development, that's crucial. Many targets aren't single, static proteins. They're big, wobbly complexes.
Which is a perfect link to our next segment. Because if you're designing one drug to hit 2 or 3 different receptors, those receptors are going to move and interact. You need cryo-EM to see that dance.
Exactly. You need to see how your drug orchestrates that whole system. But even with cryo-EM, there's still a bottleneck.
It's not the hardware anymore.
Nope, the hardware is amazing. The bottleneck is back to the biology. It's all about preparing a high-quality sample. You need incredibly pure, stable, homogeneous protein. If your sample isn't perfect, the best microscope in the world will still give you a blurry image.
Got it. So we have the design tools, we have the validation tools, now let's apply them. Segment 4. The clinical frontier of metabolic disease, GLP-1 agonists Ozempic, Wegovy have been a phenomenon. Why are they the starting point for these even newer therapies?
Because they've been so successful, they are fantastic at controlling glucose and driving real, clinically significant weight loss. But even they have a ceiling for some
And
patients.
the goal is to do even better.
The ambitious goal is to match the metabolic benefits of bariatric surgery, but with a simple injection. And to get there, a single target might not be enough. You need multi-target drugs.
Okay, let's look at the first strategy they tried, combining GLP-1 with glucagon, or GCGR, activation. On paper, it sounds perfect. Glucagon revs up energy expenditure.
Sounded amazing in preclinical studies. In rodents and monkeys, these co-agonists suppressed appetite and boosted metabolism. The weight loss was really robust.
But it didn't translate to humans, did it?
It was a classic translational failure. Human trials with compounds like co-agonists found that the big energy expenditure benefit they saw in animals just wasn't sustained in people over the long term.
So the weight loss was.
It was often comparable to just a good GLP-1 drug on its own, not dramatically better. And this is a huge point the sources make. The translational gap is real.
Any idea why?
It's complex, but it's likely our counter-regulatory physiology. The human body is just very, very good at defending its metabolic set point. You push it one way, it pushes back. The initial boost in metabolism just fades as the body adapts.
So the body fights back. If glucagon was a dead end, what about the other strategy, combining GLP-1 with its partner hormone, GIP?
Now, this is the approach that's showing incredible promise. GIP is technically an anabolic hormone. It can promote fat storage, so it seemed counterintuitive.
Right, why add something that stores fat to a weight loss drug?
Because when you combine it with GLP-1, you get this amazing synergistic effect that's more than the sum of its parts. In preclinical models, the combination led to superior glucose control and, critically, superior weight loss compared to either one alone.
And the drug that proves this concept is terzepitide.
The undisputed leader. What's so interesting about terzepitide is that its molecular backbone is actually derived from GIP, not GLP-1, and it's deliberately biased. It's about 5 times less potent at the GLP-1 receptor than native GLP-1.
So it's GIP dominant. Why does hitting the fat-storing GIP receptor harder lead to better weight loss?
The working hypothesis is that it creates a more balanced metabolic signal. The GIP action might improve the overall health of your fat cells, making them more insulin sensitive, while the GLP-1 action takes care of the appetite suppression in the brain.
A two-pronged attack.
Right. And in studies with obese mice, terzepitide produced greater and more sustained reductions in food intake and much greater weight loss than even high doses of semaglutide, the GLP-1 agonist. It seems to overcome some of that metabolic resistance.
This has been a fascinating deep dive, connecting the absolute precision of designing a molecule to the messy, complex reality of human metabolism.
It really brings it all together. To sum up, the sources show a world that's fundamentally changing. You have platforms like RFpeptides designing these incredibly complex molecules with near-atomic precision, even for targets we don't have structures for.
Right, the RBTA case.
And that new capability feeds directly into creating these more sophisticated drugs, like the multi-agonists, allowing us to optimize them in ways that were just impossible before.
So let's leave our listeners with a final thought here. We've seen that AI can design a high-affinity drug for a target that was itself only predicted by another AI. We've also seen the incredible clinical success of a complex drug like terzepitide. But we also saw that it's really hard to translate animal benefits into human results, like with the glucagon co-agonist.
It's a critical hurdle.
So if AI can design a molecule with perfect structural fit, then we know multi-agonists are the future, but are tricky to get right in humans. What's next? How soon until we see AI designing these multi-agonists specifically to overcome those known human physiological hurdles? The next frontier isn't just affinity for a target.
You're right. It's about engineering the whole system. Using AI to custom design peptides for things like oral bioavailability or stability, or ensuring that the effects we see in the lab are the same effects that last for months in a patient. The challenge is shifting from finding a binder to engineering a complete therapeutic.